what I wanted to do was to tell you about a paper which my group recently published in Heredity. So it's nothing to do with the special issue, but relevant here because it's published in Heredity and it's about plant quantitative genetics. So my next part of my talk is like a short introduction, the short story of this work we did to look at how the process of meiosis adapts to the autotetraploid state, using my favorite species to study, which is potato. So in this part of the talk, I'd like to give you a bit of background on genetic analysis or quantitative trait analysis with autotetraploid species in general. And then the study that I want to talk to you about in Heredity is about a cytological analysis of how the chromosomes behave in the autotetraploid potato and what that tells us about how meiosis can adapt to having double the number of chromosomes. So this was the paper published in June this year. So I'll start off with a really basic overview of polyploidy, because very broadly speaking, there's two different types. We have the autopolyploids, where the multiple copies of the genome come from the same or a highly similar origin. So this contrasts with an allopolyploid, where the different chromosome sets often have an origin from a different species. So here they're shown in different colors. It might be that two diploids hybridized to make an allotetraploid. So you can have a very different origin for the different sets of the genome. But what we're talking about today, because potato is an example of one of these, is autopolyploid. So an autotetraploid like potato, the 4 copies of each chromosome have the same origin. So just broadly, why autopolyploid plants are important if you're not familiar with them, not just because I love that I can put together such a beautiful slide to illustrate those, but also because a lot of plants that are important in agriculture and hence for food security are autopolyploids, and particularly autotetraploids. So I was talking about earlier in the special issue part, blueberry is an example of as an important autotetraploid fruit. And we've also got leek, strawberry, and lots of different other species here, including my favorite one, which is potato down the bottom here. So being able to understand the genetic basis of complex traits in these species is obviously more challenging with the multiple sets of chromosomes that polyploids have. So let's have a look at why that is, because what we're focusing on here is meiosis and what actually happens when you double the number of chromosomes, and I'll show you what we found in potato. So what we have here is a simple meiosis, if you like, in an autotetraploid. So we've got the 4 different copies of a chromosome in different colours here: red, blue, green and yellow. And in a diploid, it's quite straightforward because, of course, there's only one way in which the 2 copies of a particular chromosome can pair and recombine in meiosis. But of course, when you've got 4 highly similar copies of the chromosome in the autotetraploid, there's actually 3 different ways that the pairing can happen, which then creates a much broader spectrum of possible genotypes in the gametes. And you can see that here. You could have this first possibility where red pairs with green and blue with yellow, and then there's 2 more possibilities as well. And these pairings might be random, or in some species there's evidence that there's some preferential pairing between particular genome copies. So then once the pairing has happened, the recombination will then happen in the same way as it would in a diploid. Between the paired chromosomes, you get the swapping of the segments, which you can see down here, and then the recombined chromosomes will segregate into the gametes, which are of course diploid rather than haploid in an autotetraploid meiosis. So we tend to think of this as the simple case because it's very similar to what happens in a diploid, the only difference being that you've got 2 bivalents or 2 pairs instead of just 1. But of course, there's no reason why the chromosomes are restricted to that kind of behavior. It could be that they also show a quadrivalent or a multivalent, which is what you can see here. So again, we've got 4 homologous chromosomes in different colors. And here is an example where you get 4 crossovers shown by the stars here, and that holds all 4 homologs together. and it forms a ring-shaped structure. And this kind of multivalent formation, how often it occurs, is interesting to us because it actually leads to a unique outcome in the meiosis that you would never see in a diploid. And so it's important to take these things into account when analysing the genetic data of this kind of species. So if you get a crossover between a gene and the centromere, which is these black blobs in the middle here, in this ring configuration, what can happen is that when the chromosomes separate in meiosis 1 and then meiosis 2, you get the chromatids separate, you get these results. Each of these quadrants is a gamete here, and the gametes which are highlighted with the asterisk are the unique ones here. So what's happened is that alleles carried on the sister chromatids have ended up in the same gamete. And this is a phenomenon that would never happen in diploids, and we call that double reduction. So as I was saying earlier, with the different ways that the bivalents can form, this increases the spectrum of possibilities that you can get in the outcome from a meiosis. So we were interested in looking at this process of what configurations do the chromosomes show in the potato genome, because essentially we knew there were 3 basic classes of evolved autotetraploids. So we're not talking about synthetic autotetraploids that you might make in the lab, but evolved and stable autotetraploids that have been around for a long time. And what was known is that in many such examples, so Arabidopsis arenosa is an example where the tetraploidy event happened a long time ago, and over time the meiosis has become diploidized cytologically. So instead of the chromosomes forming these multivalents that I've showed you, you tend to get exclusive or close to exclusive bivalent pairing of the chromosomes. So it becomes a really diploid-like meiosis. And then in such species, often these multivalents are linked with reduced fertility because of problems segregating the chromosomes. However, it is very much a species-specific phenomenon because there's other groups of autotetraploids which have a mix of bivalents and quadrivalents, and there's even some more rare examples like cocksfoot, for example, where bivalents hardly ever occur, and it's mainly the quadrivalents or the full chromosomes forming the multivalent structure, like the ring that I showed you. So as you may know, there's a huge number of different varieties of potato, and you can see some of the phenotypic diversity here in this image from the International Potato Center in Peru. But it wasn't really practical to look at all these different varieties, but what we did is we took a selection of commercially relevant varieties in the UK, which are tetraploid, so Sante, Maris, Pia, and Cara, and had a look at the behavior of the chromosomes and the crossover frequency during meiosis compared to a diploid variety called Scapa, to see where does potato sit within this classification of autotetraploids. So what I want to show you now is what we found and what that implies about the adaptation of an autotetraploid genome. Just to show you here an image which reflects how much I love doing the cytological analysis in potato, because before this we were working with Arabidopsis, which just has really minuscule buds which are hard to work with. But as you can see here from the potato flowers, you've got these beautiful chunky anthers, which makes the cytology a little bit easier. So we can collect these anthers at different stages of development and make spreads and put together an atlas of the different stages of meiosis in the different varieties. So we did that for those 3 different tetraploids and the diploid as well. And this created an atlas which I'm showing you here. So what we did was to identify the different stages. So here we've got the start of prophase, the chromosomes start to condense and fully pair up from leptotene through to pachytene. And then when they're fully paired all the way along their length, that's where the crossovers take place, generally in the pachytene stage here. So at the top you can see the tetraploid atlas, and at the bottom we've got the diploid atlas here. So after the crossovers happen, the chromosomes, the homologous chromosomes start to pull apart, but they're held together at the sites where the crossovers have happened. And this is useful for us because we can then look at what's happening in metaphase to see where the chromosomes are held together, what shapes we can see under the microscope, and infer what crossovers have happened in a particular myocyte. So here we have all the rest of the phases through to tetrad, but it's metaphase 1 that we were interested in. So we created an uncountable number of different spreads in the search for metaphase for as many pollen mother cells as we could find to have a look at the behavior of the chromosomes and make that contrast between tetraploid and diploid. And this wasn't easy because metaphase really seems to be over in an instant in the potato meiosis and is quite hard to capture. And as you can see here in this diagram as well, often it was quite hard to get a nice clear spread where you could identify all the different chromosomes in the tetraploid. But nevertheless, we persisted for a long time and managed to get enough pollen mother cells to do this analysis that I'm about to show you. And what we did was to make it a little bit more simple rather than trying to focus on all the chromosomes, which was to some extent impossible, was to have a fluorescence in situ hybridization analysis using just 2 probes, the 5S and the 45S probes here. And this enabled us to focus on quite usefully the largest chromosome, which is chromosome 1, and the smallest, which is chromosome 2, rather than looking at all 12 chromosomes in potato. So as you can see in this diagram here, the 5S red probe here is tagging the short arm of chromosome 1, and the green is tagging the short arm of chromosome 2. So if we find myocytes in metaphase 1 and do this FISH analysis, we can then have a look at the configuration for these 2 chromosomes and infer what's going on in terms of the crossovers. And I'll show you some examples of how we do that using the images of the chromosomes. But first of all, just to give you an overview and to rationalize what we're trying to do here, what we have at the top is the really simple case, as I like to think of it, where it's a kind of diploid-like meiosis because you really only see bivalents, and these bivalents may take a rod or a ring shape generally, depending on whether you've got 1 or 2 crossovers. So here you've got the 4 homologous chromosomes in different colours like we had before, and a crossover is marked with a blue X. And these kind of crossover configurations would lead to the rod or ring shapes on the right here. So we were interested in what proportion of the time do we see this versus a whole range of other more complex configurations, including the ring that I showed you earlier, which would involve 4 crossovers holding together the 4 homologs, or chains involving 3 crossovers holding together the homologs, or maybe sometimes we see 1 homolog all by itself and there's just 3 of them held together in a chain, or a trivalent and a univalent, as we can see here. And there's a whole range of other possibilities as well, but the basic question was, how often would we see bivalent versus multivalent configurations in meiosis? So I'll show you a couple of images and how we interpreted those by using these DAPI stained spreads and the red and green FISH probes. So what we have here on the left is the DAPI stained cell in metaphase 1. And this is a really easy one, really, because what you've got here is chromosome 1 very obviously forming a ring-shaped structure. And we use the FISH. Here you can see the exact same cell with the FISH probes used as well. So first of all, we can see are the chromosomes by looking at the location, the shape of the chromosomes and the location of these red or green dots. We can infer if there's bivalent or quadrivalent pairing happening, and we can draw out an interpretative diagram of what's actually going on. So here we think this is a ring where you've got 4 crossovers shown by the stars here, involving the short and long arms of every homolog. So that one is really quite straightforward. Just to show you another example, this time with chromosome 2, here we see a chain. So we're looking at the chromosome with the green probes here, and this on the right is what we think is going on. So we've got 3 crossovers shown by the stars holding together the 4 homologs in a chain.
And why we think that is because, just to correspond the fish image with what I'm drawing out here, remember that the green fish signal is on the short arm of this chromosome 2. So this one at the bottom corresponds with the red homologue in our diagram. This kind of merged signal in the middle here corresponds with the fish signal on these 2 short arms. And then there's a third signal at the end over here for this chromosome short arm. So we're looking at the shape the chromosomes show and the location of the signals to figure out the configuration, and from the configuration we can infer what the minimum number of crossovers is that makes sense for that image. So moving on from that guide to interpretation of these cytological images, I want to show you the results and what we infer from those as to how the potato meiosis has adapted to the autotetraploid state. So what we can see here is the 3 different tetraploid varieties, Cara, Sante and Maris Pia, and the different configurations. And in blue we've got the bivalent, 2 bivalent configuration, and in red we've got the multivalents, so rings or chains. And what you can see quite obviously here is that there has been a major diploidization of meiosis in potato. So most of the time we get 2 bivalents rather than the multivalent formation. And we see the same thing on chromosome 2, which is the smaller chromosome, but in this case you see even more bivalents compared to multivalents, which is to be expected because it's a tiny chromosome compared to chromosome 1, so it's not going to have as many crossovers as chromosome 1, and it's less likely to form these multivalent configurations. But what we also saw is that there's a difference between the varieties in terms of the behavior. So in Sante, you can see there's quite a high, there was significantly higher amount of the bivalent pairing compared to quadrivalent. So what we think is that different varieties of potato have undergone the cytological diploidization to different extents, which may be linked to genetic or epigenetic features in these different varieties, which are quite different from each other genetically. So that is what we found in terms of configurations in the tetraploids. And now I want to tell you what we found in terms of how does this compare to what's happening in the diploid, which we can see in this table in the green row here. The diploid is called Scapa. So what we have in this table is the results for chromosome 1 on the left and then chromosome 2 on the right. And it's broken down into the total chromosome, the short and long arms as well. And we had some obvious results from this, really, from naive expectation, and also some less predictable results as well. So here we're comparing what's the overall frequency of crossovers in the tetraploid versus the diploid. And we found that it's much higher generally on chromosome 1 than 2, which, as I mentioned, is quite predictable because 1 is a bigger chromosome. And similarly, you get a lot more crossovers on the long arms than on the short arms of the chromosomes. But what we can add now here in green is a comparison between the diploids and the tetraploid. So the 4x chiasma or crossover frequency divided by the 2x or diploid frequency. And what you can see from most of those numbers is that it's pretty much doubled, which seems a kind of obvious result, but we have unpicked that in a little bit more detail as I'll show you. So as you can see on the long arm here, the ratio was literally 2.0. which is kind of consistent with the naive expectation because, let's face it, you've doubled the number of chromosomes. So the obvious expectation is that the crossover frequency would double, and that's that. But it's not quite as simple as that, as I will show you in the next few slides. Another interesting result is that in related species like tomato, it's been shown that there's generally no crossovers on the short arm of chromosome 2, but we found there is quite a substantial frequency given how small it is, and it contains a lot of repetitive DNA, and it's highly heterochromatic. So that was interesting for us as well. And we even saw, although it was rare, there were the rings forming for chromosome 2, just like the big ring I showed you earlier for chromosome 1. So that was slightly unexpected.
As I mentioned, we didn't focus the whole analysis on all 12 chromosomes because it was very difficult to actually get a nice spread where you could identify the correct number of bivalent or multivalent complexes. But we could do that for a small number of cells. So here, just to show you that the results were the same across all 12 chromosomes, we had an approximate doubling of the chiasma frequency in the tetraploids. But we can also see the varietal difference between the tetraploids here as well, with Sante having a much lower crossover frequency than Maris Pia, for example. So the different varieties differ in crossover frequency and also in the configurations that you see as well. But to be honest, this wasn't really a fair comparison between tetraploids and diploids because we're not comparing like with like, because as we've seen in the tetraploids, we also see quadrivalent configurations, chains and rings, for example. So what we then did is to filter the data so we're comparing like with like. How does the crossover frequency compare when you're just looking at one bivalent in diploids or two bivalent in tetraploids? So we took out the data from those cells where there's multivalents and just make a fair comparison. And this is where the results become a little bit more nuanced and interesting with respect to how the meiosis is adapting to the tetraploid state. So this is the same table as we had before, but it gives us an idea of what's going on per bivalent. So there's two bivalents in the tetraploid, so we normalize this to give a per bivalent value. And then we can compare the 4x to the 2x value as we did before. And what we can see is that the crossover frequency for our per bivalent basis is significantly less in tetraploids than it is in diploids. And that was the same on chromosome 1 as it was on chromosome 2. So basically we concluded that the per bivalent crossover frequency is reduced in tetraploids compared to diploids. And then we looked into a little bit more detail as to what's actually going on here to explain this, because every bivalent is going to have an obligate crossover so that there is correct segregation of the homologous chromosomes. And this normally happens on the long arm rather than the short arm. So what we've done here is to plot for chromosome 1 at the top, 2 at the bottom here. For each bivalent, do we see 1 or 2 chiasma or crossovers? So we can see here proportion of bivalents with either 1 or 2 crossovers. So for chromosome 1, what we can see is that in the diploid, so that's the blue lines here, we can see that these two block bars in blue are pretty much the same height. So 48% or almost half of the bivalents that we see in a diploid have a second crossover, and this is often on the short arm. Whereas if we compare the tetraploids, which are these orange, grey and yellow bars here, we see that usually there's just one crossover for each bivalent, and you don't normally see a second crossover at all. So here you can see there's much fewer, or around 28% across all the tetraploids of the bivalents actually have a second crossover. So there's this tendency for, in the tetraploids, the bivalents only will have one crossover and much more rarely will they have a second one. So the overall crossover frequency is tending towards just 1 for each bivalent. And the pattern is the same for chromosome 2 as well. So what we are seeing here in the potato meiosis is that certainly there's been a cytological diploidization because there is a predominance of these bivalents, but we do still have the quadrivalent or the multivalents being very frequent. So it varied from 7 to 48% depending on which variety we were looking at and which chromosome as well, suggesting there might be some kind of genetic control of this, which we can't really comment on right now. So we showed that the crossover frequency per bivalent is reduced in the tetraploids compared to the diploids because you just didn't see as many crossovers on the short arms in the tetraploids. So they maintain the obligate crossover on the long arm normally, but then we don't see a second one on the short arm like we do in diploids quite frequently. So we think that this indicates how the meiosis has adapted to the autotetraploid state by constraining the configuration so that we mostly have bivalents. And secondly, when you do have bivalents, by reducing the crossover frequency, which is commonly seen in a whole range of other autotetraploids as well, like Arabidopsis arenosa that I mentioned earlier. So although we haven't looked into this, we've observed the pattern but haven't actually explained it. I wanted to mention a lovely hypothesis that Kirsten Bomblies has, a nice review paper. There's a nice review paper on this which talks about the relationship between the interference in tetraploids and diploids. And the idea is that interference between crossovers involves a signal which propagates along the chromosomes, and essentially once a crossover is designated, that interference signal will reduce the likelihood of another crossover happening nearby. So maybe the scale or the distance over which this is operating is higher in tetraploids than it is in diploids. So that's definitely the kind of mechanism that we could investigate in future to try and interpret what we've seen in the tetraploids. And it's thought that the axis length is important in that as well. So further work, we have done some work on this, which I don't have time to present today. Further work is needed to look at the axis dynamics and compare that in tetraploids and diploids to see if we can gain some support for this hypothesis. So finally, this relates back to what I was talking about earlier about heredity papers in general needing to have some kind of broad take-home message. So in this case, what can we say that isn't just specific to potato? So we think that constraints on meiotic chromosome configurations are an important feature of an evolved autotetraploid meiosis in general, but it can show up in different ways in different species. So it can be quite a specific phenomenon. What we think is going on is that because potato is a largely clonally propagated species, maybe it hasn't been under the same pressure as other wild or autopolyploid populations. So it hasn't evolved a meiosis that's strictly dominated by the bivalents, and it still has a high proportion of the multivalent pairing as well. And that is important to know because when we're doing quantitative genetic analysis in autotetraploid species, as I showed you earlier, if we've got a high frequency of multivalent formation, so for example, the rings, this could lead to double reduction, which is a unique genetic outcome in the myocytes. So taking into account this complexity, we believe is important in doing genetic analyses such as QTL analyses in this kind of species. So thank you very much for your time and attention. I just want to finish with some acknowledgements here. So all of this cytological work was done by my PhD student, Anushree Chowdhury. Potato material was provided by Ankush Prashar in Newcastle, and myself and Eugenio were involved with interpretation of the cytological images along with Anushree and Jing Shen, who is now a postdoc at UCL, and Patricia Ponce, who is my PhD student, also helped with the interpretation of the images here as well. And we had a fantastic undergraduate student who spent the whole summer searching for metaphases, Liam Wright working with us on this project, and fantastic technical support from Steve Price as well at the University of Birmingham. And finally, last but not least, we thank BBSRC for supporting this work. And I will be happy to answer any questions.
Thanks very much, Lindsey. Yes, and it looks extremely challenging to identify those shapes within those crossovers there in the squashes. That does look quite challenging, I must admit, especially the numbers that you got to, for sure. So.
Yeah, absolutely. Anushree was an absolute whiz at this, as is Eugenio. And I've learned how to do it myself for sure, but sometimes we'd sit down with these images and Anushree explained it to me and I just couldn't even see it. Sometimes it is really challenging to do these things. So I showed some nice simple examples here, but it's not always that straightforward.
Right, yeah.